was like, was I ever like going to get any? Yeah. We could go.
Probably not. Exciting. It's kind of cool.
I feel like I'm going to get
Well, anyway, you honestly, this day is fun.
You guys found this stuff?
What's up?
Is that one of the presentations?
Yeah, that's one.
Okay. I did have the slides with all of them because some of the presenters, not that many, but like some guys.
Yeah, I think everybody is.
no
Yeah.
question.
Okay, yeah, so just be sure.
Yeah, this year, no, I haven't changed because this year, every year is 1 hour. This year is all hours. So we assigned it for that.
So what should we do?
But it's still 50 minutes per person.
Yeah, total. So basically the alarm should ring at 12 minutes, and that's a max. And then if they, if you have 3 today, okay, if there's really some people who have 3,
Yeah, yeah.
then you
Quite
say
recent
no.
deviation. That's good.
Yeah, but it's always 15.
Yeah, next one all. So it's just
I know.
yeah.
Yeah.
Or,
But if
no,
I wouldn't be upset if I like.
Oh, no, I mean,
if came
the
back.
15 minutes extra.
It would be.
Okay, that's a good question because we have extra time built in here.
Yeah, well,
Yeah, I know.
so,
Right. So last
Kind
year,
of under,
if we always had to be, and we had so many this year,
but I know this.
I see.
we had it.
Because the transition time.
I was like, just do
Yeah,
it
so
for the week.
then I just kind of have a little.
Yeah.
My heart.
I'm just gonna.
Thanks.
I was like, I was assuming you were.
Yeah, do you think I can email Amy?
Do you want to go load your presentation up?
I think it was at one point at least. I'm not sure if it still is.
Yeah.
If you want to go ahead and just make sure, yeah.
Are we doing it like all of them are just popped up and we can
Yeah,
go down here and grab it?
or I'll like, in
Okay.
between, I'll pull it up for you.
Yeah,
And I'll- Okay, cool. And
here's
then
mine. I could put my name on it if that would make it easier for you.
It's, I'll know.
Okay. Yeah, that one.
Okay, and then Tahida, the timekeeper should be sitting right there. And then December 15 minutes per presentation. She'll give you a 3-minute warning at 12 minutes.
Okay.
And then a 1 minute.
And that's including question time, right?
Yeah. But I mean, the session is a little bit longer.
Okay.
So we had some that session. That's
Because I should aim for like 10 to 12 minutes on the actual talking.
yeah, and like whatever you want for Q&A. You know,
Okay.
if you want less, talk more.
It's never a problem. Always easy to do. Yeah, I think I hit 12 minutes on Saturday. But they had a big clock right before right in front of the podium. It was amazing.
Angle, perfectly angled.
Yeah, so from, if you're sitting in the crowd, you can't see anything at all. It just looks like there was like a, you know, like when they hang like things off the tables. So it looked like it was just like something like that in the floor. But on this side, it was a TV that had like a little bit of your slides, you know, a giant clock that was counting down. So it's very good.
Exactly. Thank you. Yes, that's exactly what I was thinking.
Hey, Chris.
Yep.
Okay, sorry, so we sorted out the timing a little bit. It will be closer to 12 minutes. So you'll have 15 minutes.
Okay.
Guaranteed. But our session, 4 speakers. So if you try, if you're more than 12 minutes, okay. Okay. There'll be some issues in the total time for that transition. If you just try to get around.
About one.
But yeah.
There were like a few times it was like 11 minutes and 30 seconds on the dot. Like, Yeah, I was expecting it to vary a little bit. I was like, I hope you're not robots, but.
Feeling inspired? Yeah. That's great. I'm David. I think that study is busy. I think we are. That's great. I was at, I'm a, I was at, you were representative of strength on a really, really good one. Good.
Oh, no, it's not. Sorry.
Make the most of it.
Diamond-based? No. But it was a picture that I saw.
I only had one of those presentations. What was that? Oh, sure.
All right, we'll go ahead and get started. Sorry, I was waiting for our fourth presenter, but I don't. Are you her? Oh, okay, great, you're here. Okay, yeah, I got it up here. Okay, we're good to go. Okay, so we'll go ahead and get started. You are in the session for microbial risk and treatment, so if that sounds right, then you did okay. And today, we'll kick it off today first, First, my name is Kevin Zhu, and I'm the moderator for this session. But first, we'll hear first from Christopher Anderson from West Virginia University. So please give Chris your attention. It's off. Okay, you may have to be very direct into the microphone.
Okay, I can just try to speak loud. Okay. All right, hello everyone. So as you mentioned, my name is Christopher Anderson. I'm a first-year PhD student at West Virginia University. And today I'm going to talk a little bit about wastewater-based epidemiology within combined sewer systems. So a little background with combined sewer systems. Nearly 860 communities in the United States are served by combined sewer system infrastructure that collect both domestic sanitary waste and stormwater. And particularly these are common in the Rust Belt and the Appalachian regions, as well as in small communities and larger communities in that area. The common problem with combined sewer systems is the idea of rainfall-derived inflow and infiltration. So this is a thing that's hard for operators as well to manage with their capacity. But because of this rainfall-derived inflow and infiltration, either through cracks in pipes in their infrastructure or directly by collecting rainfall, they struggle with this all the time. And particularly areas with aging infrastructure such as Appalachia are more prone to issues with rainfall-derived inflow and infiltration. And the reason we care about this is because wastewater physiochemistry is known to change during periods of increased RDII. So specifically your nutrient loadings, your organic loadings, commonly they're diluted. And so that makes us ask the question, what impact does RDII have on wastewater epidemiology? And this is especially important when you consider regions that have disadvantaged populations, such as some areas in the Appalachian region. And you can kind of consider this nexus of healthcare proxies, such as wastewater epidemiology, that can inform for those disadvantaged populations in terms of access and health. But as part of that, you also have to understand the infrastructure characteristics that are common to those disadvantaged populations as well. And so in order to provide an equitable wastewater epidemiology to some of these regions, we need to kind of tie them all together. There have been studies that address sampling in terms of like what types of samples we might take, grab versus composite, when to take samples, and just in general, what makes up a wastewater sample matrix when it comes to these viral targets that we're looking for. But oftentimes, these have not considered wet weather impact at the forefront. And so our study had three primary objectives that we addressed within combined sewer systems with the wet weather impact built into our study design. And so for the sake of time today, I'm only going to focus on the first one, which was to compare diurnal fluctuations of viral RNA in wastewater treatment plant influent during dry versus wet weather conditions. And so to do this, we selected two different facilities, both of which are combined sewer system, or have combined sewer system infrastructure. The main difference between these two was that facility A was larger in terms of its flow capacity and average flows through the day, as well as its customers that it serves and the extent of its sewer network. But something really important to pull away from this that we found interesting was that facility B had an I&I, or an inflow and infiltration rate, of about 77% over the 2022 calendar year, which essentially points to the fact that they are receiving more non-fecal water than they are domestic sanitary waste in the influent at their treatment facility. As far as sample collection was concerned, we developed this kind of parallel autosampler configuration in which we had one autosampler that was programmed to collect a 24-hour composite collecting samples hourly throughout that 24-hour duration and combining them. And then the other automated sampler was programmed to collect those as segregated, discrete grab samples throughout the day. So we could then later compare the efficiency on dry and wet weather of composite samples versus grab samples. But for what I'm talking about mostly today, we are focusing on those hourly grabs to look at those diurnal fluctuations. And so in doing this, we sampled each facility for 3 dry weather days, not having any rain, and also 3 wet weather days having rain. And then as far as the lab work goes, we sampled for a number of wastewater physiochemical variables, including chemical oxygen demand, ammonia, suspended solids, pH, and about 4 or 5 others. And also, we sampled for, or analyzed for SARS-CoV-2, COVID-19 viral fecal shed RNA, and pepper-mild model virus, which is a human fecal indicator because of its abundance in the human gut microbiome. So, as far as some broad results are concerned, here you have facility A and the days showing the PMMOV concentrations on dry and wet weather. So the yellow squares on this represent the concentration of PMMOV throughout the day. Also, you'll notice in the kind of the background of these figures, the white shaded region represents a monthly average dry flow that we calculated using all the dry weather days in each respective month we sampled to kind of give us a baseline of how those flows changed on during wet weather. And so as part of that, the green shaded region is the actual recorded flow at this facility. We also noticed something interesting at facility A while we were collecting samples, which was these septage discharges, denoted with the triangles on this figure. So basically, because of the extensive decentralized treatment in the area, trucks will frequently go out to septic systems and pump them, but then they are bringing them back to this facility and dumping them in the influent anywhere from 3 to 8 times a day from various regions and parts of the state throughout the weekdays. So to kind of pull this together, we had 3 key points that we wanted to express with this. So the first of which was at facility A, the wet weather flows were significantly higher than the monthly average dry flows. And so this essentially is telling us that, okay, these rainfall events did cause these drastic changes in flow. And as part of that, point 2, PMMOV and SARS-CoV-2 were both significantly lower during wet weather, which hints at the fact that these are becoming diluted with the presence of rainfall. And then point 3, septage discharges may actually cause elevated viral RNA concentrations. And so that observation that we actually had evidence shown here of where a truck, the time ticket we received for it was between 9:53 and 10:02. We pulled a sample at 10, and so our sample was directly compromised by that septic truck and might not have been representative of the community we were actually sampling. As for facility B, we have the same figure, so I won't explain it again, but the kind of key points at facility B, things were very different at facility B. And so first of all, the wet weather flows were not consistently greater than the monthly average dry flows. And there was just this general inconsistency with flows throughout the sampling at facility B. So typically with wastewater influent flows, you would expect kind of a sinusoidal-like diurnal curve that's evident, but most of the flows at this facility were pretty constant throughout each individual day. Secondly, SARS-CoV-2 was significantly lower during wet weather, but there was no significant difference in PMMOV. And this basically told us that the SARS-CoV-2 was more likely to be the change in SARS-CoV-2 was more likely to be due to prevalence changing in the community in terms of infection, as opposed to a change due to the weather criteria. And then third, the high rates of I&I, if you'll remember, it was about 77%, reflected consistent dilution of viral targets regardless of dry or wet weather. And so all of these concentrations were likely lower than they actually would have been considering the influent infiltration. So kind of a few more things just to pull it all together. We also noticed that minimal rainfall, oftentimes less than 1 inch, caused dilution to a point of near non-detection. And so you can imagine if we're designing this study based on weather, right, we essentially become storm chasers to an extent. And at that rate, it was very difficult to get storms that were of high magnitudes. But even still, with very minimal rainfall over catchment areas, we did observe these dilution effects. We also noticed a first flush of viral RNA that occurred pretty frequently among these samples. So with the onset of a steep rise in the flow hydrographs, this, a few times at least, coincided with a temporary peak in the viral RNA abundance, followed by an elongated period of dilution throughout the rest of that elevated flow. Interestingly enough, we found that peak hourly viral abundance only coincided with peak flow rate on one occasion. And so in the literature up to this point, a lot of people have used the assumption that when flows are highest, that is indicative of most people using the restroom or flushing water. But in combined sewer systems, that's not necessarily the case. And so we concluded that, at least in this study in combined sewer systems, the peak flow rate did not coincide with peak fecal loading and therefore did not coincide with peak viral abundance necessarily. And lastly, the diurnal patterns of viral RNA loading are less evident when subject to elevated rates of I&I, regardless of dry or wet weather. And so the big takeaway from all of this work was that the ability to accurately detect and quantify viral RNA targets during wet weather events is highly dependent on the foundational characteristics of each system. And so the need then becomes to define both these systems and their waste streams in order to interpret the wastewater epidemiology in an accurate way to inform public health intervention moving forward. And so that is all I have. I'd like to thank a few of my funding sources, as well as all the members of the Watch WP team, our surveillance group, my PIs, and my undergrad, Alex, who helped me with a lot of the lab work in this. So I'm very thankful to him because it was a lot of lab work. And I can only get into the surface level of this, but I have a lot more data. If anybody would like to contact me by email or talk with me afterward, feel free. And also feel free to visit our surveillance dashboard and our social media page. And that is all I have.
We'll now take questions.
How do you?
If you have a question, please.
So my name is Molly Osa from Emory University. So I've only recently gotten into wastewater surveillance, so I apologize if this question makes zero sense. I was just wondering, so you're talking about how like rain and heavy rainfall events might dilute the presence of viral detection in wastewater. Do you know how that would change if you are using like the liquid methods versus solid methods to detect viral load in wastewater?
I don't necessarily know how that would change, other than the fact that, of course, suspended solids are also very dilute when there's rainfall. So if you're relying on compacting those solids again, say collecting sludge at a wastewater facility, it is possible that those are getting flushed through treatment quicker. We didn't look at that specifically, but I do know that there have been studies that show the viral targets can be more concentrated in those solids. So we did look at the liquid fraction, though. Yep.
A similar question. So one is about, you compared, it's really useful the way you've compared different methods of sampling. I don't know if you've tried more swabs or other kinds of passive sampling? And also I was interested if you tried using crucified or some other marker, because I guess with the model virus you may have environmental sources. If you tried a more human-specific marker as well instead of that one on that.
Yeah, so we didn't try any other markers. We kind of selected PMMOV somewhat as a baseline, but also just in general as a virus. And for our purposes, you know, it was kind of as simple as like, well, how did it change from dry versus wet weather, and was it dilute or was it not dilute? So those environmental sources are definitely interesting, but we did not get into that as much. And then your other question was what again?
All this sort of solid state
Oh,
samples. Yeah.
and we also did not get into passive sampling. Our big thing with some of the other objectives we looked into were kind of, when comparing those two, which I would love to show you this later, was kind of getting at time windows to take these things. So whether you're taking a grab sample in the afternoon hours or the morning hours, or how representative, how those match up during dry and wet weather. And we noticed that so because of those periods of dilution that are throughout the latter part of a day, maybe, right? If you take a grab sample, then it is not going to be even representative of what you would take in a composite because you're kind of missing your opportunity due to those changing flows in the dilution. But we did not try passive sampling, though.
Cool. Thank you, Chris. That's all. Next up, please welcome David Holcomb from University of North Carolina.
Afternoon, everyone. I'm going to be talking about fecal indicators a little bit today and what they can tell us about exposure to specific enteric pathogens in children. So there are a lot of different pathogens that we might care about in the environment, might want to assess exposure to them in environmental matrices, but they can be really challenging and expensive to detect comprehensively and especially quantitatively. So a typical approach is to instead look for more abundant microbes or other constituents of human feces, or feces generally, that might represent a proxy for exposure to these pathogens. And in particular, there's some thought that human pathogens might be of a greater infection risk. And so we might care more about whether the pathogens are coming from human feces specifically. And so we can look at human-specific fecal indicators or markers that may be indicative of human-source pathogens that may be of a greater risk. So in this work, we wanted to estimate associations between human-associated fecal indicators and general fecal indicators with exposure to enteric pathogens in child stool. So specific associations for each pathogen as well as looking at an overall effect for a typical pathogen. So we did this as a nested study inside of the Maputo sanitation trial, which is a study that was conducted in Maputo, Mozambique. We did 2 cross-sections for this work in 2015 and 2016 that were focused at the compound level. So these are household clusters that share outdoor space and is where children tend to spend a lot of their time and would experience most of their exposures. So we enrolled children that were less than 4 years old at baseline in 2015 and then had a second survey in 2016 where we concurrently collected child stool samples and samples of household stored water and soil from the entrance to the compound latrine and to each household that had an enrolled child in the study. And so with these 3 different sample types that we collected for the stool, we used a PCR-based method to detect bacteria, protozoa, and viruses, and then also used microscopy to look for soil-transmitted helminth eggs. With the environmental samples, we used membrane filtration to concentrate them, and then qPCR to look for 2 different human-specific markers, HF183 and MNIF. And then we also quantified gene copies of the E. coli 23S gene and cultured using MTEC for E. coli colony counts as a non-specific fecal indicator. And then we used a modeling approach that allowed us to consider all of the pathogens that were assessed at the same time in the same model, looking at associations between exposure to each of these different fecal indicators in the different, in the water and soil, looking at the associations between exposure to those indicators and detection of each of the pathogens in stool, as well as a pooled association across all the pathogens that represents what you might think of as a typical pathogen, what you might expect from that. So we had slightly different numbers of stools that were available with matched environmental samples depending on the PCR-based detection of pathogens versus the microscopy. But we had just shy of 200 stools with matched samples. With the water samples, we had multiple child stools that associated with water, but for the soil, since there were more samples collected at each compound, we actually had multiple soil samples associated with each child. So the sample sizes differ a little bit here. So overall, what we found for the stored water, we estimated the impact on prevalence of each of these individual pathogens for a log 10 increase in the concentration of E. coli or for detecting one of the human markers because they were detected less frequently. So each column here represents one of the fecal indicators, and then each row is a specific pathogen sorted from highest crude prevalence to lowest. So that's what's in the parentheses here. The null at 1 means that we didn't really see any effect detecting the fecal indicator didn't have any association with the prevalence of the pathogen. And then less than 1 means that we find lower prevalence of the pathogen, above 1 is a higher prevalence. And for water, we really didn't detect the second human marker MNIF at all, so that's not going to be reported here. So overall, we really just saw one clear association, which was when you have increasing gene copies of E. coli in stored water, we found a higher prevalence of Ascaris in the child stool. However, there were a number of pathogens that appeared to be lower prevalence, so it's a marginally significant, but was pretty consistent when we detected the human marker HF183 in water. These were all the most prevalent pathogens, so some of them were detected in over half of the stools, but the human marker was only detected in about 15% of the water samples, so there might be some artifact arising from that. Interestingly, these same most prevalent pathogens all were marginally higher. The prevalence was marginally higher when we had increasing E. coli gene copies as well. And so this is reflected in this pooled association, which we think of as reflecting a typical pathogen, where none of them were a very clear pattern, but the pathogens overall were a little bit elevated with increasing E. coli gene copies, but a little bit lower prevalence with HF183, but definitely consistent with a wide range of outcomes there. For the soil, one thing to note is that all of the indicators were detected much more frequently, including the MNIF this time. And there was just a larger sample size overall, as I mentioned earlier, with more soil samples. But generally, there was just no clear pattern at all across all of the pathogens for any of these indicators. The one bit that sticks out a little bit is a suggestive increase in Campylobacter when we detected HF183 in soil. But overall, there really was not any clear patterns that we detected here. So we certainly have many limitations with this work. The big one is that the exposure, these fecal indicators in household environments were assessed at the exact same time as the outcome, the pathogens in child stool. So this is basically assuming that these exposures were constant back in time when the exposure would have happened, which is not ideal. And we've seen in previous work that if you do a prospective ascertainment of the exposure for, say, E. coli in water, they have found stronger associations with diarrhea and other symptomology later. That's assessed later on. We also just had a fairly limited sample size, although we were looking at many outcomes at the same time here. Also, the typical pathogen, what we're considering this pooled association that we're estimating, is still dependent on which pathogens are included in the analysis, and so it really reflects typical of what we have included here, but would be affected by including other pathogens that might have other transmission pathways or biology. And then the human markers that we assessed here just have really poor specificity and sensitivity in this setting in particular, and it's pretty common in very densely populated domestic contexts generally, that they tend to not perform very well. And so there's just limited information coming from detecting any of these. But overall, it seems like human markers are not a very reliable indicator of exposure to specific pathogens in this setting. We also don't really see consistent associations with E. coli, although the increased ascaris prevalence in water with increasing E. coli was interesting, and mirrors what was seen in the wash benefits Kenya trial, where the drinking water treatment arms found lower prevalence of ascaris in children's stool. But overall, it seems like direct pathogen detection is probably a more promising approach. It's still expensive, but it's much more possible now with the multi-pathogen platforms like TACMAN array cards that are available. So we'd really like to thank our partners, the National Institute of Health in Mozambique, and the implementing partner for MapSan overall, water and sanitation for the urban poor. We appreciate our funders, but they are not responsible for any of what I have said here. And I would be happy to take any questions. Also, please come and see other MapSan sessions at the conference. There are several here, especially the side event that we have on Thursday, where we'll be explaining more about the trial and how it was designed and implemented. So thank you very much. Yeah.
Hi, great presentation. I was just curious, and I might have missed it, but did you do analysis of like any pathogen altogether combined by taxa, or did you just do the single pathogen analyses?
We just did the single pathogen analyses. We didn't want to do the combined outcome because it's sort of a different question. It's asking, have you detected any one of these? Which is interesting, but we were really interested in whether there were associations with specific ones, and then if we could look at sort of an average over all of them. Yeah, but that's certainly something we could also look at.
Yeah, I mean, I think that would be interesting.
Part of the reason we didn't do that is that the prevalence of any pathogen is near 100% in this cohort. So you have to then start making decisions about what to leave out.
But potentially grouping them by taxa.
True. Sorry.
This is really interesting. How is the role of animal contact within these compounds considered, since most of those pathogens were zoonotic?
Yeah, so we also, we mostly had poultry in these compounds. There are dogs and cats as well, but poultry were what we were concerned with, and we used the GFD marker and almost never detected it in any sample, so weren't really able to analyze it. There certainly were animals in the compounds, but they didn't show up in the source tracking information.
Yeah, I was just curious, so the non-detection of HF-183, is that suggesting non-human exposure?
It could be that, it could just be that HF183 was not a good marker in this setting. So the sensitivity and specificity were both about 2/3. And then with the water samples, you know, we weren't able to concentrate enough maybe to really detect it. My guess is that there's still a lot of human feces moving around here. Yes?
I'm excited about your association with drinking water quality and ascaris infection. Maybe I missed this, but you haven't looked for ascaris in water yet.
We did not in this round. We are currently in the new round of MapSan looking for it in water using a custom TAC. So more to come on that. Actually, during the side event, there will be some information on that. Oh no, there won't be. We're not there yet. I take it back. All right, thanks so much.
All right, please welcome Abul Kamal from International Center for Diarrheal Disease Research, Bangladesh.
Good afternoon, everyone. I am Abul Kamal from Environmental Health and Wash Unit, icddr, Bangladesh. So today I will present one of my study titled Strategy to Control Antimicrobial Resistance in Wastewater Treatment Plant Effluent from Municipal, Hospital and Domestic Wastewater. A systematic review. So there is an increased prevalence of AMR in the environment, and it caused a significant public health concern. Untreated wastewater is an important reservoir of antibiotic resistance bacteria, antibiotic resistance gene, and antibiotic residue. And untreated wastewater usually contained various sources like human, animal, and environment. Wastewater treatment plant plays a crucial role in disseminating ARB, ARG, and antibiotic residue before releasing to the environment. There is an increase in AMR research in recent years. However, a lack of synthesis of evidence on the effective treatment method for removing ARB, ARG, and antibiotic residue in the wastewater treatment plant that receive municipal, hospital, and domestic wastewater. So, our objective was to synthesize the current evidence on the efficiency of different new and conventional wastewater treatment methods for lessening the level of ARB and ARG in the effluent, evaluate the presence and concentration of type of antibiotic residue in the effluent from wastewater treatment plant, and to determine if wastewater treatment process promote the selection of ARB despite an effective decrease in the total number of bacteria. So in our study, by new treatment method, we mean that those treatment methods that provide integrational alternative for the treatment of wastewater for reducing contamination, and the conventional wastewater treatment method, we mean that the treatment plant system with a combination of process including physical, chemical, and biological method for removing solid and organic matter. We published our protocol in JMIR Research Protocol Journal, and then we followed the PRISMA guideline. The data source we used Ovid Medline, Ovid Embase, Web of Science, environmental collection from ProQuest, and then probe. And the time frame we used from 1st January 2020 to 30 December 2020. The reason behind the time frame limitation has been added since study related to AMR published prior to the 2001 did not have an environmental focus. This is the Prisma diagram, sorry. And you can see that at the, from the all database, we initially found 6,569 articles, and after the duplication removed, we actually screened title and abstract of 3,892 articles, and then 974 article, full text article was assessed for eligibility, and at the end, we include 34 study for our analysis. For objective 1, 26 articles met the inclusion criteria. For objective 2, 10 articles met the inclusion criteria, and for objective 3, only 3 articles met the inclusion criteria. Regarding the geographical distribution of study included in the systematic review, you can see that most of the articles included from United States, followed by study conducted in Poland, Italy, France, and then some high-income and low-income countries. We also assess the risk of bias. The risk of bias tool was adapted from the previously published tool by William Nguyen et al. 2016. You can see that in our study, overall risk of bias, most of the study has high risk of bias, and followed by around 13 or 14 studies, they have unclear risk of bias. In the domain of sample selection bias, you can see that around 6 articles showed low risk, around 14 articles shows high risk. Regarding the domain of information bias, most of the articles showed low risk. And in the confounding domain, most of the articles showed unclear risk. Effectiveness of wastewater treatment plan reducing ARG. 14 study measure ARG from 18 individual wastewater treatment plant. Conventional treatment plan were used majority. 4 study measure ARG from 5 wastewater treatment plant where new method were used. The new method were ultraviolet radiation, substance filter, and biological agent filter. The conventional treatment, here you can see that, here you can see that conventional treatment plant with new method showed the higher removal rate compared to the conventional treatment plant alone. Regarding the effectiveness of reducing the ARB, out of 20, 18 study measured ARB from 19 individual wastewater treatment plant, 17 were conventional treatment plant and 2 were conventional treatment plant with new method. And the new method were activated carbon filter and multimedia filter. The pool removal rate was higher in the conventional treatment plant with new method, and it was 98.42%. Regarding the efficiency of removing antibiotic resistance, you can see that 8 studies were included for meta-analysis out of 10. In 8 studies, there were 10 wastewater treatment plants, and the new method were used in 4 wastewater treatment plants, and the new method were ultraviolet radiation, multimedia filter, and activated carbon filter. Irrespective of wastewater treatment plant types, the pool removal rate of antibiotic from the wastewater treatment plant of 8 study was 12.11%. Here you can see that in some study, 1, 2, and 3, these 3 study, the antibiotic residue was not that good compared to the other rest. In summary, we can say that conventional treatment method with new, conventional treatment plan with new method show better removal of ARB and ARG. However, the antibiotic residue, the result is mixed. This is because for some antibiotic, the removal rate is good, but for some antibiotic, the removal rate is poor. Even the treatment plan use new method. That means that for the wastewater treatment plant, antibiotic residue depend on the type of antibiotic. Some antibiotic remove goods, some is poor. Regarding the selection of ARB, only 3 studies mention selective pressure, wastewater treatment plant process, and cell carrying resistance gene may cause selective pressure. So our study do have some limitation. One of the limitation is that it is not possible to determine which technology is better than other. High level of heterogeneity was observed across the study. There is a lack of information on whether the study followed standard guideline for antibiotic susceptibility testing. There is a investigation of bacterial AMR that rely on culture-based method may underestimate the actual number of AMR. There is no standard procedure to measure antibiotic concentration from wastewater in different country and time. So the study was funded by Swedish International Development Cooperation Agency, Sweden, and this is our review team member, and then this is my contact details if you have any question. Any question, please?
Hi, Kamal. Thank you very much. So I'm in the same team, but I'm not aware about this project, sorry. So, Kamal, I'm just curious, you mentioned about that you don't have any understanding about the wastewater treatment plan for the AMR removal. But there are many studies have been actually conclusive that they have some effect on removal of AMR genes. So also we did one hospital-based study that the systematic review suggested that there are some wastewater treatment. So have you ever have any like wastewater treatment based on membrane, which is very effective removing this? So I don't see any of them have membrane-based wastewater treatment plan.
Thank you very much for your last question. Yes, there is some wastewater treatment plant, they actually use membrane. I forget to mention one thing in our study that we only include wastewater treatment plant that receive municipal, hospital, and domestic wastewater. And if the wastewater treatment plant receive industrial wastewater or pre-treated wastewater, we did not include in our study. Because the industrial wastewater, the things contained in the industrial wastewater is not biologically degradable. So I think the treatment plant we found in our systemic review, they did not use membrane filter. Thank you. Anything else?
All right, for our last speaker, please welcome Temitope.
No, you're good. It's perfect. Yeah. Hi, everyone. My name is Temi, and I'm a PhD candidate at North Carolina State University. And today I'm going to be talking about exploring the use of next-generation sequencing for characterizing environmental transmission of antimicrobial resistance. Okay, so antibiotics are one of the most significant medical breakthroughs of the 20th century, if not the most significant medical breakthrough. And yet the development and spread of antimicrobial resistance threatens to undermine their effectiveness and poses a serious public health threat. In fact, in 2019, the Centers for Disease Control estimated about 2.8 million people contracted AMR infections in the United States, and more than 35,000 died from their illness. The environment has also been identified as a key pathway of transmission to humans, and so it's really important that we understand how AMR spreads in the environment and how it further develops. And so E. coli has one of the highest rates of resistance in bacteria. In fact, the World Health Organization designated extended spectrum beta-lactamase, or ESBL, producing E. coli as a public health threat that required sustained and prompt action. The WHO also underscored environmental surveillance as a crucial step in understanding and combating the rise of antibiotic resistance. New surveillance tools are needed to detect and characterize AMR pathogens such as ESBL-producing E. coli and their spread. And so whole genome sequencing has shown great potential for epidemiological surveillance, where culture methods identify the presence and concentration of a target microorganism. Whole genome sequencing gives detailed genomic information that can be used to characterize AMR microorganisms of interest, such as ESBL-producing E. coli, and it can also be used in conjunction with wastewater surveillance. And so previous studies have highlighted the use of wastewater surveillance for monitoring the spread of antimicrobial resistance, as researchers have been able to detect new forms of resistance and undetected or unreported AMR pathogen outbreaks. Given the widespread development of wastewater infrastructure during the COVID-19 pandemic, it's imperative that we leverage this resource for AMR surveillance. And so typically, whole genome sequencing is conducted on a single isolate or a metagenomic sample. With single isolate sequencing, you're able to get deep and even coverage of your microorganism, and assembling the genome afterwards is often straightforward and pretty simple, but there are downsides to this method. It's limited in the contextual genomic information provided, and working with environmental reservoirs requires a clear snapshot of the microbes present since they interact and influence one another. Metagenomic sequencing is an alternative method to single isolate sequencing. There's no culture method, and so you're, in theory, able to capture all of the microorganisms present in your sample. But there are drawbacks to this method as well. Genome assembly requires an evenness and depth of coverage that can be difficult to obtain for all organisms in your sample with this approach. And the random nature of metagenomic sequencing may lead to a high representation of irrelevant pathogens, and you may miss clinically relevant AMR pathogens that are present at a low concentration. And so mixed culture sequencing addresses the drawbacks of both of these methods. A mixed culture is a cultured sample with various microorganisms, typically of the same genus, present. And the benefit of working with a mixed culture sample is the ability to analyze multiple strains of a target microorganism that's present in an environmental sample, while getting a depth and even coverage across the genome that isn't always possible with metagenomic sequencing. This is due to the smaller variety of microorganisms present in a mixed culture sample. Additionally, using culture ensures that all of your organisms phenotypically express antimicrobial resistance. And so I developed a genomic surveillance system that will allow sequencing of ESBL-producing E. coli isolated from environmental samples. And so I'll be analyzing ESBL-producing E. coli found in 2 wastewater treatment plants located right here in North Carolina. So the research objectives are just 3 as follows. The first is to develop and validate this method for sequencing multiple strains of ESBL E. coli in the environment. The second is to characterize the diversity of ESBL-producing E. coli present in wastewater over time using this technique. And the third objective is to compare the diversity of antibiotic-resistant genes, or ARGs, and ESBL-producing E. coli strains within our 2 wastewater treatment plants and identify any potential temporal variations. So we collected 85 wastewater samples from 2 wastewater treatment plants in Wake County, North Carolina. Wastewater treatment plant 1 serves most of Raleigh, which is our capital, about 30 minutes from here. And wastewater treatment plant 2 serves a suburb of Raleigh, and it also captures the Raleigh-Durham International Airport, which most of you probably flew in through. And so we collected 24-hour composites of wastewater twice a week from January 31st to June 27th of 2023. And so here's a brief overview of the genomic surveillance system that I've developed. I'm going to go ahead and give an overview of it, and then I'll go into detail on a few key steps. So we started off with culturing our wastewater samples for E. coli, or antimicrobial resistant E. coli, using a modified IDEX assay, which I'll talk a little bit more about in a minute. We then extracted the DNA from the sample and prepared the samples for sequencing and used a long-read sequencer, specifically the nanopore MinION. We then did some pre-processing of the generated reads, and then we did de novo assembly with Metafly. And then we performed an additional polishing step. And so I'm going to go ahead and focus on these three pieces of the surveillance system: the culture method, the sequencing, and the genome assembly. So we cultured samples using a modified IDEX assay that I helped to develop in the Harris lab. Briefly, 10 milliliters of wastewater was added to 90 milliliters of deionized water, followed by 5 milligrams per milliliter of cefotaxime, which is an antibiotic. And resistance to cefotaxime typically indicates ESBL production. This was followed by color 18, which is a media created by IDEX to culture E. coli and total coliforms. The culture that was positive for presumptive ESBL-producing E. coli was then used and taken throughout the rest of the surveillance system. So as I mentioned, we did this on wastewater samples, but we also piloted this pipeline on bacterial isolates, specifically ECNC11, which is an ESBL-producing E. coli reference strain, EC29522, which is a non-resistant E. coli reference strain, and Klebsiella pneumoniae, which was a non-resistant total coliform. And we also created a mock community made up of these three bacterial isolates to confirm the ability of the pipeline to work to sequence E. coli and total coliforms. For sequencing, we decided to use the Oxford Nanopore technology MinION, as it's a highly portable sequencer that can generate reads as long as 100 kilobase pair long. Long-read sequencing is optimal for a mixed culture like this, as it reduces the complexity of genome assembly. And the MinION also provides better resolution of plasmid DNA, and many ARGs exist on plasmids. This feature allows for better characterization of AMR microorganisms overall. And we use the ligation sequencing kit for library preparation, as it's Nanopore's most high throughput kit. For de novo assembly, we use Metafly, which is a tool designed for long reads. It assembles and polishes our contigs. And then we performed additional polishing with Medaka, because Nanopore reads often have higher error rates. And so for some of our results, these are the results from the bacterial isolates and the mock community experiments. And as you can see, we were able to identify each isolate individually, as well as in the mock community. Additionally, we use StrainGE, which is a metagenomic strain separation tool to quantify the amount of E. coli strains in our samples, and it accurately identified 2 strains of E. coli in our mock community. We were also able to detect multiple antibiotic-resistant genes that confer resistance to antibiotics such as penicillins, cephalosporins, and fluoroquinolone antibiotics. And here are a list of a few of the antibiotics that we noticed resistance to. And here's some of our preliminary wastewater data. These 2 plots represent a taxonomic classification of some of our wastewater samples. And as you can see, E. coli is the dominant species in each sample. As I mentioned earlier, the modified IDEX assay cultures for E. coli and other total coliforms. And we're primarily interested in ESBL-producing E. coli. And as you can see, that's what we're predominantly getting in all of our samples. We also detected numerous ARGs associated with E. coli in our samples, and this heat map just displays a few of them. And we detected genes that encoded for multi-drug resistance as well. And so now while this is a very preliminary analysis, because we're still optimizing our bioinformatic pipeline, using StrainGE, we did identify on average 4 distinct E. coli strains in each of our wastewater samples on average. And here we have a few beta-lactamase genes that were detected in our wastewater sites. Many of these genes encode for resistance to important penicillins and cephalosporins. And this further confirms the ability of our pipeline to sequence ESBL-producing E. coli. We also detected numerous plasmids, many of which contained ARGs. Plasmids are largely responsible for the spread of antimicrobial resistance, since they are mobile genetic elements, and many of the plasmid-mediated resistance genes found in pathogens originate from environmental habitats. We also detected many virulence factors associated with E. coli in our samples, which point to the pathogenicity of many of the strains that we are detecting. And so just a few current limitations. We're currently working on a method to generate full genomes of the individual E. coli strains. Currently, we've been able to extract 1 E. coli genome from each sample, but we're interested in being able to construct full metagenomic assembled genomes for all of the strains available. Additionally, culturing bacteria can promote bias, and so to address this, we plan to conduct an additional mock community experiment where multiple strains of antibiotic resistance E. coli will be combined at various known concentrations and cultured and taken through the entire genomic surveillance system, and this will allow us to assess sequencing-based quantification. And just some anticipated implications. This method is a novel technique for the surveillance of ESBL-producing E. coli found in the environment. The use of culture and sequence gives us in-depth information on this clinically relevant pathogen in wastewater. Additionally, the modified IDEX assay works with other sample types such as feces and soil, and so this pipeline is highly adaptable. Additionally, the assay also works well in low and middle income countries as it's incredibly portable. I don't know if many of you have seen a MinION sequencer, but it weighs about 100 grams and is smaller than my palm. This method also does not require extensive research infrastructure such as autoclaves or biosafety cabinets. And as a result, this technique holds the potential to enable real-time surveillance of antimicrobial resistant pathogens in low and middle income countries. And with that, I want to say thank you to everyone that contributed to this project and to you all for listening. And are there any questions? Yeah.
I'm just going to ask about you talked about the bias.
Mm-hmm.
The bias might be a good thing in some ways. If you're biasing for organisms that are more virulent or more culturable, it might not be a problem. So don't necessarily assume a bias is a problem. You might be able to, just look at critically at the bias.
Yeah, that's a great recommendation. I'll definitely keep that context in mind. Any other questions? Yes.
Can you talk about interpretability of results with this method,
Yeah.
metagenomics, and how it compares to enrichment methods?
Yeah, that's a great question. So many of the pipelines and tools that we're currently using, as I mentioned, this is preliminary data, so we're still in the process of optimizing, but there are tools that were developed for metagenomic samples, but often, like StrainGE, for example, was for low complexity metagenomic samples. And so with a sample like this, where it's a mixed culture, there's multiple strains of E. coli, and as you saw, other total coliforms, but it's primarily that. It's not as wide array, or it's not a high diversity as a metagenomic sample. We're essentially able to use some of the same tools, but get more relevant information to AMR and enteropathogens specifically. Does that answer your question? Yeah.
Thank you for your presentation. You said you use the modified IDEX assay culture method. I guess this is different from the IDEX method we know we use for E. coli contamination. Is the modified IDEX assay available?
Yeah, so the modified IDEX assay is more so a protocol than a system. So we just take a typical IDEX, all the reagents that they provide, such as the media and the trays, and the only difference is we add cefotaxime. And so we just validated the use of adding antibiotics to an IDEX system to quantify and enumerate antimicrobial resistant E. coli. And so if you have all of the materials already for an IDEX assay, the only additional thing you would need was to purchase the antibiotic, and then you could use a modified IDEX assay to culture antimicrobial resistant E. coli.
And then my second question is, I work in a water lab, and we just started AMR surveillance in the environment. I think both you and the earlier presenter looked at this at the wastewater treatment plant. Have you also looked at the AMR in the environment?
That's a great question. So that's actually one of the follow-up projects that we're hoping to do this December, where we'll be traveling to Tanzania and using this system to detect AMR E. coli in surface soil, animal feces, human stool, as well as stored water, drinking water, et cetera. And so we plan to use this method on different environmental reservoirs. And because we validated the use of the modified IDEX assay with those reservoirs, we expect that to work, I guess.
So
Yeah.
can I invite you to also come and do the same work in Uganda?
That'd be really cool, but you have to take that up with my advisor. But yeah. Oh, sorry. Yes. Hi.
This is really great work, and thanks so much for the excellent presentation.
Thank you.
I was wondering if you could say just a little bit more about what you're trying to do to get whole genomes.
Yeah.
I mean, it seems really challenging.
Yeah.
Those samples, so I'm just curious if you could say a little bit more about that.
That's a great question. So right now, one of the things that I mentioned as next steps is that additional mock community experiment where we'll take different strains of ESBL producing E. coli, culturing that at different known concentrations, and then taking that through the pipeline. We plan to try out multiple different metagenomic strain separation pipelines on those samples to see which works best, and then going from there. And so it's a little bit of trial and error, which I guess is a lot of research anyways. But yeah, we're planning on using that to just determine what would work best for our samples, because there's so many tools available. But yeah.
I think your time is up.
I was going to say, I don't know if I have a time.
Just one question,
Yeah.
that are you recommending that we don't need to go to the lab to estimate ESBL producing bacteria if we use this tool?
Well, not,
What is the, like, have you tested this in the laboratory settings versus your portable one?
yeah, so that's a great question. So right now with this system that we're using, we get our wastewater delivered to us and we are using this in a lab, but we have used the modified IDEX assay in the field. And so the hope is, like I mentioned, we're going to be in Tanzania this December, is that this pipeline would work in a lab, like how we're using it here in North Carolina, but it would also work in a field laboratory setting. Does that answer your question?
I'll talk to you later.
Okay.
We have more time for questions. This is the last presentation.
Oh, okay. Benefits. Any more? Yes.
Can you just go back to where you talked about the sequencing of the mid-ion?
Yeah.
I might have missed it.
No worries.
Yeah, just, so what were you sequencing, like, what were you targeting?
Yeah, that's a great question. So we were using whole genome sequencing. So we just had the culture, any culture that was positive for presumptive ESBL producing E. coli. And if you've used the IDEX system, you see it's just whatever fluoresces blue under UV light. We then aliquoted some of that culture, created a cell pellet, and then extracted all the DNA from that cell pellet, and then took that sample and used that as the input for the mid-ion. And so the goal was whole genome sequencing. Yes. Yeah, that's a great question. So in the beginning, when we were still optimizing the protocol, we were initially going through well by well and taking it out. But then for the sake of this project specifically, we weren't so much interested in quantification. And so we would just leave it in the vessel. So we would, put it in the vessel and, usually you transfer it over to the tray and then seal it and then put it in the incubator. We just left it in that 120 milliliter vessel, put it in the incubator, and then shined it under UV light, made sure that we were seeing that it fluoresced blue, and then took an aliquot from there. But in the, no, in the beginning we were, but we were like, we're not so interested in quantification, and it is a bit time consuming to go through the 59 wells, especially if we're not using the quantification data. And so you can use the wells. We did use it and it did work, but we didn't use that for most of the samples or any. Great. Well, thank you guys so much. I really appreciate it.
Thanks, everyone. That concludes our session today. Thank you. Too good. I know. Okay, see you.
We know about IDEX, same thing, new device for I'm not sure this is the first
Oh, yeah. I'll bring you by the tech room and we can ask. So you emailed it to someone, right? Yes. Yeah, okay. I'll walk you over to the tech room and then we can find somebody to answer the question. Right now? Yeah, or if you want to hang out and ask a question, I can wait around. From here, I just don't want you to get lost. So, yeah, I'll go. Maybe you just show me. Okay, yeah. But if you want to stay around and ask, I'm not. Okay.
and that's the one that Stool is.
Stop training. What's that level already?
Yeah, sorry, and so that's kind of why I was asking that, because that's what I wanted, that was one of
today, like, yeah, well, I mean, just the things due to like several of us, there's a lot of things got, a lot of things got collected and yeah, and the laboratory is not analyzing what kind of things happening in the lab. It always happens, right? So I went over to postdoc and then the data came in, and then I started seeing you years. I was afraid that I like but it's not, right? It's just so much easier. Now I have the animal it was in the animal system. Really? But I've been like, when I come back to it, I've been like, this is thinking, I was like, I actually have the data from the compounds with the observation.
because we're controlling our respiratory pathway so well, it's you know, we call it wind vomiting disease. What would you think of it?
Especially if it's across our people, so like the kid is already infected with it, and then you're going to the water. Yeah, not even, yeah, just. That's just like
It shows that it's a different level of training pathways in different communities.
some of the AR ones when they're at yeah, just like hypothesis.
With that data, they for that type of target at the end, at a facility, then it's a little more clear cut. Respiratory viruses and things like that.
But yeah, you said you were
had that in the children case that don't care facilities hit by wind.
One use of just research, to me, we're always sure that when I try and if I ever do kind of any kind of modelling type or it's fun, because often a real lack of number of the data. And the wastewater grants data is often the data we've got for wastewater monitoring data is often the best data we've got for just the levels spread and season. So we might not know what they use it for, but knowing that this is a way of getting campaigns we use, the Dutch have done a commercial researchers can access it, they can see and ask questions. So I don't think we should be afraid to just say that we want to just know because we can do it, therefore we're going to do it.
Yes, and you have to also be ready for the cows of like, if you're spending money, you don't have like and goal and just making it clear that the end goal is that this data set will help serve gets expensive.
It is the kind of thing we use it for. It's not directly linked to an immediate habitat section. And norovirus, I don't think we're doing active handling norovirus suicide. Knowing we'd be able to specify why how different patterns and other different goals. For example, the work that colleagues back around the Tim was doing, they were showing how immune appear into the US, and 6 months later, and then show up, it doesn't come through. It's useful to see. We didn't act on it, but it was useful to know. So they said, okay, we're going to get norovirus this year because it's 4 years since the last one got tried. Here will be gone. It's now in the US. We're going to get it. It's going to be crap. So you would come from each of those companies.
Yeah. The action might not be as direct or.
No. I suppose it could be for vulnerable groups. You could maybe do something like groups.
Like diagnostic clinical surgery, kind of have a warning through your local community wastewater that would tell you that this is surgery and trigger the appropriate test. If you were going to test for anything, to try to rule out something more serious.
You also might get a really virulent kind of coronavirus one year that severe symptoms, for instance, an immune overreaction or something like Spanish flu or something. And knowing in long-term history how norovirus is disseminated in the community in different populations, and they're spread around the world and so on. That could be useful if we get a very nasty type emerge.
Do you see commercial interests ever wanting to use it? Like if you were a cruise ship planner, would you ever test members before onboarding to try to help?
It's a good question, because that's the classic cruise ship process. I think they're a bit cowboyish, that's my opinion. They might not want to live not want to know, because you think they could be testing people. They could be testing wastewater diseases, that's what's on the cruise ship. You could do it, you could get a test on board on the cruise ship. I'm not sure they want to know. It's a good question. I was talking to them. They have an international health regulation. They're big businesses. They might be interested in talking about what they can do if they're starting to see it rise. But strictly speaking, they should be implementing strict measures all the time.
I don't know if there's ever lines with their business interests, like if they're actually like the whole cruise ship is hit by something, they're doling out a bunch of refunds. They have this like prevention, like
I suspect they'd be told you should be doing all the preventive measures. You should assume that those infected anyway. It's part of the practice. It's worth asking them if they're finding this for the people on board find this.
the commercial component is something that I mean, it's one of the reasons why I can't like do or testing or research and there's platforms that actually supply commercial for the testing. But then there's also commercial market that won't do that testing. So one supporting like biotech that does the testing and then supporting
We had a lot of Australian mining companies using it, American mining companies operating with it. We had, what was it, this based in Texas? Chevron, I think. Chevron, they were using huge storage at the university for them. They were using that, and we developed method. Training for PhD. They're testing the mining camps, and they're finding it really useful. There's like SARS-CoV-2 free mining camps, and they just keep getting races all the time. And they found it, they could lock it down, test all the individual mining huts, and finally had it isolated using that test. Because it was flying fire. They found it useful.
Now that you said that, I should link on the biggest one. The NBA is because it's a ton of money for those, playoff games and everything. And so they actually have like a bubble for that first year for the playoffs where, like my friend of mine was assistant coach on one of the teams that went in, and he had just had baby number one. And his wife was like, you either win everything or you lose like the very first round of the playoffs when they went all the way to the final. And they didn't let anybody work for anything, so they were just like.
I know Japan was in the Olympics with it as well. I bet they had a bubble system.
Yeah, there's probably a lot of noise. I don't think that's that level of lockdown, but what I'm saying is there was incentive to have testing.
I don't know, you know, not maybe that's probably what I'm dying, but if you've got an NBA team, you wouldn't want to have to against any kind of promotion or anything. So if you can have a way of getting early warning, that would be really useful. But I guess if it's a small team, you just testing. You're living in a community, you can see. Hey Rory, what's in this community where we're living at the moment? What's sort of out there? Oh, we've got this new strain of influenza that's just come in earlier than normal. We're going to affect so many people, we're going to get it, or maybe move them, put them onto strict measures because it's now circulating, we're going to get it. And they could find that useful.
A certain Fecal or Fecal is on positive testing.
It's not only like virus infection or pandemic, but also like vaccine disease samples. We're also collecting the disease data, chemistry, and we're trying to integrate when the wastewater is signaling increase, what kind of disease. Also like climate data, like temperature, rainfall, humidity.
That's something like most of it didn't have time to go into, but as far as like based on physical chemistry is concerned, we looked at a lot of common parameters and it was, I want to say 8 or 9 total, like common wastewater, like quality markers that are really about like treatment facilities. Kind of so that maybe you can determine when you take a sample, maybe you're looking at the treatment facilities data to say, okay, they usually have high CP at this time, and regardless of
Like correlation is very difficult. In our town, because it varies like seasonality, temperature impact. So you have to have a continuous system to understand the pattern. Otherwise, it's very difficult to conclude what should be the next year plan. So that's why I think it should be continuous. It should not be like 3 months, 6 months. Thank you very much. Best of luck. PhD is like a part-time.
Cut it down to 12 slides, 10 minutes. And you can use that same data. Well, the same thing with writing a paper.
like, especially most of the grass is like, I guess there is like behind the buildings. Yeah.
Thank you so much. I appreciate what you said.
They spent like so much money and time to I guess like
This is, yeah.
Oh, me too. I'm from Philly. Where are you from? Sorry, I get so excited. Oh, it's like 15, 20 minutes.
yeah, it's all like
Trying to keep
yeah, or like across the street.
So I was like, yep, I need to remember.
Yeah, they're all.
But
Yeah.
I am
Oh, yeah. They
very
basically
close to
got rid of
submitting it to
every single parking spot they had, and it's buildings. And then they've been trying to put in that bus depot for like 3 years, which just seems like you just paid for some reason and have been out yet.
And that was the other thing is I've thought about like, should I break this up? Okay.
I know. I feel like I'm just here for it's like, now when I do analysis like that, I just think like maybe it has a pool.